This section, we will learn Neoplastic Atypia. First, we learn the definition atypia. The term refers to the extent to which neoplastic cells differ from normal cells, both cell cytologically and tissue structurally. Atypia have implication for the diagnosis of tumors and for decisions regarding their treatment. Another term, differentiation. Differentiation means the parenchymal cell resemble the normal cell they are derived. So the differentiation focus on the similarity, but atypia means the parenchymal cell, the tumor cell, differ from the normal cell. Two aspects: structure and cellular structure and tissue structure. Differentiation can divide four subtypes: well differentiated, moderately differentiated, poorly differentiated, undifferentiated. Well differentiated cells are morphologically the same or very similar to the normal tissue. This appearance suggests a benign, non-invasive, or less aggressive tumor. Poorly differentiated cells differ markedly from the normal tissue, vary in size and shape. Their origin is often difficult to recognize without the aid of immunohistochemistry or electron microscopy. So, if the tumor is very poorly differentiated, we need to do some special stain. For example, immunostaining. Poorly differentiated cells are characteristic of malignant, invasive, aggressive tumor. Now let's see this picture. There are three pictures. This is the normal gland, one layer, one lumen. This is the well differentiated tumor tissue. This tumor tissue is similar to the normal, and this is the poorly differentiated tissue. We can find the neoplastic tissue are different from the normal. So this is well differentiated, very similar. This is poorly differentiated. They are different. Another term, anaplasia. Anaplasia means hyperchromasia, high ancillation. Usually we can easily find the tumor germ cell. So anaplasia show hyperchromasia, high ancillation, easily found tumor germ cell. The last word, pleomorphism. Pleomorphism means prominent variation in size and shape of tumor cell. Now let's see this slide. This is an anaplastic tumor. We can find the cellular atypia in anaplastic tumor of rhabdosarcoma. We can find the marked cellular and nuclear pleomorphism, hyperchromatic nuclei, and tumor germ cell. This is the tumor germ cell. And we can find the cellular structure and shape are marked different from show marked cellular atypia or cellular pleomorphism. This is the multinucleated germ cell. Okay, so the size, the shape are different. This is the anaplastic tumor. Second part, we will learn the contents of atypia. The content of atypia, there are two aspects. The first is cytological atypia. The second is tissue atypia. The cytological atypia contains cellular pleomorphism, nuclear pleomorphism, and the change of the cytoplasm. The nuclear pleomorphism is the most important. This is the key feature to determine a malignant tumor. Now let's learn cytological atypia. Cellular pleomorphism, nuclear pleomorphism, and the cytoplasm change. There are three aspects. Cellular pleomorphism, a term used to describe the extent to variation in cell size and shape. So there are two key points. The first is cell size. The second is cell shape. In malignant tumors, some cells may be extremely small and primitive appearing. Some cells may be many times larger than neighbors, even from the tumor germ cell. The second aspect for cytological atypia is nuclear pleomorphism. This is the key points for determining a malignant tumor. Nuclear pleomorphism describes the variations occurring in nuclear size, shape, number, nuclear membrane, mitosis, and nucleolus. So if you want to observe nuclear pleomorphism, you need to observe these key points: size, shape, number, membrane, nuclear membrane, mitotic figures, and nuclei. Size. Nuclei are often larger than normal, so the nuclear the nuclear to cytoplasmic ratio may approaching 1:1. The normal is 1:4 or 1:6. Shape of the nuclei usually variable. Nuclei are often hyperchromatic, so the staining is very dark because of abundant DNA. The nuclear membrane becomes thickened, and the chromatin may be clumped. Sometimes there are two nuclei, even multiple nuclei, then form joint cell. Let's see this picture, this slice. This is the rhabdosarcoma, a highly malignant tumor. In this slice, we can find very big nucleus, multiple nuclei, and the chromatin are clumped. The nuclear membrane becomes thickened. In malignant tumor, they are often numerous mitosis. So mitotic figures in malignant tumor are often bizarre, abnormal. So we call this is a pathologic mitotic figure. So we can find mitotic figure, but this is in the normal tissue we can find mitotic figures too. But the mitotic figures are the structure of the mitotic figures are normal. The pathologic mitotic figures can be asymmetric, tripolar, quadripolar, or multipolar spindles. Some mitotic figures are so abnormal in shape that they are termed abortive. Now let's see this slice. This is the tripolar mitotic figures. This is abortive mitotic figures. This is abortive mitotic figures. Mitotic figures, we can find the mitotic figures. The key pathologic changes is the nuclear membrane disappear, and the staining is uniform. It's very dark blue and it's uniform. So the shape for the mitotic figures is irregular. This is mitotic figure. This is mitotic abortive, tripolar. The third aspect for cytological atypia is changes of cytoplasm. The cytoplasm become basophilic, so the staining for the cytoplasm is purple, and the cytoplasm is decreased. Sometimes we can find the abnormal products or secretion, for example, mucus, glycogen, lipid, pigment. These abnormal products or secretion can help to determine histogenesis of the tumor. This is the malignant tumor cell. In the cytoplasm, we can find the mucus. This is another slice. This is the mucus in the cytoplasm. So this is the mucus in the cytoplasm. This is the nucleus. The nuclei are eccentric. So we call this is the signet ring cells. This is the melanoma. In the cytoplasm, we can easily find the melanin. So cytological atypia contains three aspects. The first is cellular pleomorphism, the second is nuclear pleomorphism, the last is the changes of the cytoplasm. The second part for atypia is tissue atypia. Tissue atypia refer to differences between neoplastic tissue and the normal tissue in structure. There are three key points. The first is cell arrangement. The second is polarization. The last is the relationship between stroma and parenchyma. Benign tumors usually differ only slightly from normal tissue. Malignant tumors usually have a highly disorderly structure in terms of cell arrangement, cell layers, and the relationship of parenchyma cells to the stroma. Now let's see this slice. This is the normal squamous epithelium. The normal structure for squamous epithelium. There are many layers. The surface is keratin layer. The bottom is basal layer. The surface is flat. This is a normal squamous epithelium. How about this one? This is the squamous cell carcinoma. We can find the tumor cell form nests. The keratin layer in the center, and the basal layer is in the surround. They are different from the normal. The normal keratin layer is on the surface. The basal layer is on the bottom. So they are different in tissue structure. This is squamous cell carcinoma. This side is the normal epithelium. The surface is the keratin layer. The bottom is the basal layer. Okay, the surface is flat. How about this one? This is the squamous cell carcinoma. The structure is different from the normal. We can find the nest. This is a nest. This is a benign tumor derived from squamous epithelium. And this structure is different from the normal epithelium. We can find the finger-like structure. This is a finger-like structure. Finger-like structure. And this is another picture. We can easily find the finger-like structure. So in the tumor tissue, we can find the tissue atypia, both in benign tumor and malignant tumor. This is another example. This is the normal glands, normal intestinal glands. The normal structure is, we can find one layer. This is just one layer, only one layer. And this cuboid cell form a glandular structure, just one lumen. This is normal structure, one layer and one lumen. How about this one? This picture are different from the normal. Here we can still find the one layer, one lumen, but this slide, this is the parenchyma and this is the stroma. This is a normal parenchyma. This is the stroma. The stroma become decrease in the neoplastic tissue. So this is the adenoma. Now let's see this slide. This is a normal glandular structure. And now let's see this picture. We can find that there are many. We can find that there are many layers. There are many layers. Okay. In the normal glandular structure, just one layer. There are many layers. And here we can find the many lumens. So the structure of the parenchyma are different from the normal. Okay. This is the parenchyma and this is stroma. So the tissue atypia or the cellular atypia refers to the parenchyma are different. The parenchyma components are different from the normal. Now let's see this size. This is the normal. This size is normal. One layer cell, one lumen. Here, many layers. So this structure are abnormal, are different from the normal. And the lumen are irregular. So this is the normal, this is the tumor. Now let's see this slide. This is a normal structure for the colon. And this is the adenoma, shows slight atypia. This is the adenocarcinoma. We can find the prominent atypia. Many layers. Okay, the lumen are irregular, or many lumens. So in this section, you need to master the definition of atypia and the differentiation. Two definitions. Second, you need to master the contents of atypia. There are two aspects: cytological atypia and tissue atypia. Okay, that's all.